I'm Mitchell Volger, and I'm a postdoctoral fellow at University of Washington School of Medicine. I currently work with Andrew Sturgakis, but at the time of doing this work, I was in Evan Eichler's lab in genome sciences. To quote a very wise person from the department, Debbie Nickerson, variation is the spice of life. Without variation, the field of genetics would be extremely boring to study. So getting a more complete understanding of how we differ and potentially how that impacts our phenotypes is very important and easy to motivate, I think, from a genetics perspective. If you look at sort of the regular type of a variation that's easy to study, usually you see about 5 million base pairs where you get like single base pair differences between individuals. But if you look at copy number variation and other types of variation where you get sort of large segments of sequence that have moved, you can get tens of millions of bases that vary between individuals. So a lot of this new sequence we uncovered last year were in these copy number variable regions where there's lots of differences between people. So my focus in this latest work was taking the 100 haplotypes that we assembled in this latest effort and looking at these variable regions and discovering the additional diversity that exists in there and starting to characterize that. Depending on how you choose to count, definitely with the way I choose to count, the vast majority of human variation comes from these copy number variable regions that are only going to be unlocked through the use of a pangenome reference. So I think it's absolutely critical that that we continue to push this resource and that the community begins to adopt it.